So welcome back. Thank you for sticking with us and joining us here on day three of Common Fund Data Ecosystem CFDE Pediatric Power Up. I'm joined again with my co-instructor Sangeeta, but today is going to be I'm going to be leading most of the lesson today. And we're right here in the middle. We're in day three, so we're already approaching the halfway point of our course. And so to kind of review and set context of where we've been and where we're going. The first 2 days have been kind of informational. So we've learned a lot about what types of data sets are out there through the Common Fund data ecosystem and these DCC organizations and how they make their data available through these different platforms. And then yesterday we had a great lesson about different tools and methods in bioinformatics. So types of experimental strategies that folks can follow and some introduction to tooling that you can use. And then today we're going to dive in and we're going to do an analysis today. So we're going to do an RNA-seq analysis using a software tool called DE-seq2 that's already built out for us in this cloud platform called Cavatica. And so we're going to be using an existing tool and we're going to load up some data and we're going to set it up so that it's able to run. And then we're going to look at some output as well. And then looking ahead to tomorrow, so Sangeeta's going to take the mic back tomorrow and she's going to show us a slightly more complex example where she's going to be building her own pipeline and building her own workflow. So I know folks in our chat were describing how they use their own tools, they use their own pipelines. And so, this is an opportunity to show how you can do some of that in the cloud, in this cloud platform, Cavatica. And then Friday is going to be a very kind of open day. So it's going to be, Sangeeta and I, and we believe some of our partners from Valsera and Cavatica are going to be here as well. To help answer questions and connect you with resources to help take your project to the next level. So we're right here in the middle on day three and looking ahead to stuff at the later in the week. All right. So day three, so what are we covering today? So I want to provide a brief introduction to this concept of RNA-seq since it is going to be a feature of what we're talking about today and tomorrow. Cover some of the high-level principles of the analysis and what we're going to be looking for. Then we're going to navigate to the Kids First portal, and we're going to select some files for our analysis. We're going to then move those files into Cavatica to run a analysis pipeline using the software tool DE-seq2. And then we're going to show you two different ways that you can incorporate GTEx expression data into your analysis at the end. All right, so let's get started. So to review, what is RNA sequencing? So RNA-seq is derived from the high throughput sequencing technologies, a lot of which Sangeeta introduced yesterday. And so they work, I used to say they all work the same way, but now we have new long read technologies which work differently. So we already have the next generation after next generation. So that's why I like to call these just high throughput sequencing because they make data available much more rapidly than was possible through traditional Sanger sequencing methods. So step one of the analysis is you take your DNA molecules that you're trying to sequence, and you fragment the genome into tiny little pieces, and then run those through what's called a flow cell. So it's shown here on the bottom right. It's these two plates of glass that are very close together. That there's a channel that runs through them. So the molecules run through the channel and get stuck, and so you get these little short molecules of DNA strands affixed to the glass. And then by washing different colored nucleotides through the flow channel and then taking pictures and tracking how the colors change, we can read, the order of the base pairs at each of those individual spots. So each of these spots on the glass becomes what's known as a read. And so a read is shown here. This is a collection of reads. This is two reads in what's called a FASTQ format. And so this is just reading out the output of a FASTQ file head, and then this designation .fastq. And so you can see we've got names for these reads, kind of an identifier, and then this is the actual sequence, the GAAGAN, and then there's even an N, so the sequencer wasn't sure exactly what base pair that was, so on and so forth. And then, so this corresponds to 4 lines. There's a quality score down here, and then the whole thing repeats itself once again with another read. And this will continue for perhaps millions of reads, depending on the depth of our sequencing that we're trying to work on. So each of these FASTQ files, these are what are called unaligned reads or raw reads. So these come right off the sequencing machine. These are very big files, and they're not very useful in this state. So at this point, we don't have any information about where these pieces of DNA interact and connect with each other. So remember, if we go back to our previous slide on the glass, the DNA is completely fragmented, and so these two pieces, just because they might be next to each other on the glass plate, doesn't mean that the sequences are near each other in the genome. Those could be on 2 completely different chromosomes. So likewise, the reads in the file, there's no kind of organizational information about them at this point. We don't know if this read and this read are anywhere near each other. And so the analogy I like to share is that it's a bit like solving a puzzle. So when we get our reads from the right off the sequencer, you can see that the reads are a bit like these puzzle pieces. So when you have a box with a puzzle, the puzzle pieces are all mixed randomly together. There is no organization to them at that point. Whereas, so then what we do is as you take the pieces out, you look at them, you find which ones have, similar colors, and you piece them together in order to be able to build out the puzzle. And you can see this is sea creatures, in this puzzle on the diagram here. And so that's a bit what we're doing when we do this genome assembly, is that we're finding these puzzle pieces, these reads, and finding which ones look similar and overlap, and then building them together into the genome of the sample we're looking at. So I see a question in the chat. What's the difference between Sanger and high throughput? Great question. So Sanger sequencing is an older technology that has been around in a couple of different iterations, but it was used prevalently in the 90s as the kind of the beginning of the Human Genome Project. And so you're building these relatively longer reads. You get about 600 or 700 base pairs, but it's much harder to do it at scale. So you get really high quality results for a single area, a single part of the genome, but it makes it very, very hard and very, very slow if you're trying to sequence, the entire genome of an organism. So there's slightly different kind of biochemical techniques that go into it, and I'll allow you to follow up if you're curious to learn more about the chemistry of the techniques. But the overall differences is that the Sanger technique is really good if you're trying to sequence a single gene. Maybe you have a candidate gene and you want to sequence it in many different samples. So that might be the best sequencing strategy for you to choose. But if you're interested in kind of whole genome work and gene discovery, you want to search the entire genome to find things that might be disrupted or have variants in them, then you would want to choose, an experimental strategy like high throughput sequencing, which is capturing the entire genome and allows you to do it much more kind of quicker and rapidly. All right, so we've got our box of puzzle pieces here. So we need to assemble these reads using these terms into what's called a genome alignment. So reads, so a bit like our puzzle pieces. So here's our puzzle pieces, kind of represented more closely as DNA here. And so just like putting together a puzzle, you can imagine this is the picture. This is the complete picture, and these are our pieces. And so how the software works is that it looks at these individual reads and tries to find regions that are similar to one another. And so you can see, this TATGGC at this read looks a lot like TATGGC on this second read. So these pieces look similar. Let's plug these pieces together. And so you can see here they are, and there's the overlap. And so now we have a slightly bigger, piece of the puzzle. Then likewise, oh, the end of 2, GCGTAC, looks a lot like GCGTAC in number 3. So we can attach that, and now we have 3 pieces put together. So just like we're putting together a jigsaw puzzle, on our dining room table, we can also jigsaw together these reads into longer DNA contigs. And so this is called aligned reads. So we're taking the reads and we're aligning them to this reference genome. And so we're putting together the puzzle. You can imagine the puzzle's a lot easier if you have the box. You can look at the cover of the box and say, oh, this is the sky. I'm going to put, these blue pieces together. Oh, this is a tree. I'm going to put these green pieces together. And so we're aligning them to the reference. These files, have usually a .bam or more recently a .cram extension. These are, I call them medium big, so they're a little bit smaller than the raw FASTQs, and they have useful information. So we're now starting to represent, this is what, a more complete picture of what the genome looks like. We don't have, puzzle pieces in a box anymore. We have an assembled puzzle on our table. So today, though, so that's kind of an intro to the principles of this DNA alignment. But today we're specifically looking at RNA sequencing. And so to talk about RNA sequencing, there might be a couple different reasons why folks are interested in choosing RNA sequencing as their experimental design. But the big thing that we're looking for with RNA sequencing, and that most people are looking for, is that they want to capture gene expression. So I'm going to take us all back to biology 101 with the central dogma. So remember, in our nucleus, all of our cells have DNA. DNA is copied into DNA itself again by DNA polymerase. And then when genes are being expressed, the DNA is transcribed into RNA. And then that RNA floats out of the nucleus and finds a ribosome. And then it's translated into protein. And then those proteins have specific shapes, which influences characteristics of the organism or the cell. And so you can imagine that a gene that is highly expressed in a particular tissue will have a high amount of RNA from it, and then also a high amount of protein from it. And so this RNA, so we're going to have lots of, the DNA quantity is going to be the same, but we'll make more RNA if it's highly expressed. We'll go through this middle arrow quite a bit, and then we'll have more protein, as a result downstream. So what RNA-seq is doing is that we're trying to quantify the amount of RNA molecules in a particular sample in order to calculate rates of expression. So if a gene is really highly expressed, then it's going to have lots of RNA, and a gene that's lowly expressed will have few or no RNA. And so if we can count the amount of RNA for those two genes, then we can compare. Or likewise, if we have two different samples, we can compare expression by comparing across those two. Let's see. So we see, does the CRAM format include aligned sequences? And Sangeeta is already popping in. Yes, thank you. So, the kids' first CRAMs include the unaligned pieces as well. So it does include the unaligned reads in addition to the, so you can imagine we have a complete puzzle, and then there's a couple other pieces that don't fit in the puzzle. Those are included at the bottom of the CRAM file. So at least in terms of the kids' first data. And then Valerie appreciates the jigsaw analogy, so that's great. So Bejan brings up this point, cDNA, so that's a great point. So because RNA, if you've worked with RNA before, you'll know that RNA is a really kind of fickle molecule. You need to be very careful if you're working with it in the laboratory. You need to spray everything down with ethanol. You need to use special pipettes and special tips because it's such a precious molecule. It degrades very, very rapidly in the environment. There's lots of RNases that will chew it up. So likewise, we can't feed RNA through that glass plate and run it on the sequencer. So we actually, the first step is to reverse transcribe our RNA back into cDNA and then run that cDNA through the plate. But again, because we're doing it on scale, the amount of RNA for each gene, if we have lots of gene A, and then we run the whole thing through reverse transcriptase, then we'll have lots of cDNA for gene A as well. So that's our kind of overview of where we're going here and kind of or what I've been describing in more of a pictorial format. So we want to compare the expression of two genes in two different samples. So here is a collection of RNA that collected from sample 1, and here's a collection of RNA from sample 2. So we'll generate reads and align them back to the reference genome. And not kind of included in my directions here is that we would take this mRNA after isolating it, and we would reverse transcribe it into cDNA and then run the cDNA through the sequencer. And then we align it to the genome. And so now as we have these pieces, so we would expect, again, because the RNA only comes from genes, so if we align it to the genome, we're going to have big parts of the genome where there's no reads mapping, but then the reads themselves are going to line up with those specific genes. So we'll see that with sample 1, here's the genes reads corresponding to gene A, and here's the reads corresponding to gene B. And then likewise, sample 2 is gene A and gene B. And so we can estimate expression based on the amount of coverage, or what's called read depth, within those particular genes. So if you compare sample 1 to sample 2, you see that gene A has higher amounts of expression than gene B. It has more reads than gene A. And so we can say, well, gene A has 30 in sample 1 and has 10 in sample 2. So assuming we, fed through the same amount of RNA to begin with, and assume that we're doing proper controls, we could estimate that gene A has a 3-fold higher expression in sample 1 than it does in sample 2. Likewise, with gene B, we see that we have, 10 over here for gene B and 5 over here. So we have a 2-fold change. So we can test this, by looking at these different kind of controls. We can look at and estimate relative fold changes. Due to different lengths, it may need normalization. Yes, another great point. So genes are not all the same length. So some genes are massive and some genes are teeny tiny. And so you could expect that, a massive gene is going to have a lot more molecules just from the sake of the fact that it's so much longer. And so that type of normalization will occur as we as part of our software tool. So it will take into account expression across the entire genome, factoring in, well, how much expression do we have in all genes in order to be able to estimate and normalize. Is the size of the gene important in counting the number of RNA? Yes, so it is significant, but the software tools will be, kind of accounting for that. And so there's a metric, this kind of reads per kilobase. And so it, accounts for, a kind of a proportion. So yes, thank you, Wenshen RPKM. So reads per kilobase. And so that allows you to kind of do a normalization for genes that might be longer or shorter than other ones. All right. So that is my kind of intro talk. So what we're doing and what we're looking at. So what we're looking at today is we're going to find we're not going to do an alignment, and we're not going to do, so we're going to kind of start a little bit further downstream. We're going to grab this gene quantity expression counts. So we're looking at these tables of genes, and we're going to be doing an analysis where we're just going to be looking at the output of that alignment and calling workflow. So we're going to identify some RNA-seq expression files on the Kits First portal. We're going to push those files to Cavatica, our cloud analysis platform. We're going to set up the DESeq2 app. We're going to run it, and we're going to look at some output. And then I'm going to show you how we could combine with some expression data from GTEx. So that's kind of our guiding stars and what we're going to try to work on over the next about hour and 20 minutes or so. Excuse me, hour and 40 minutes or so. All right. So now I'm going to step away from my slides, and I'm going to minimize these. And I need to readjust some of my windows to get going here. And I think we're ready to get started. Okay. So we are going to get started. So I guess I'll share broadly the directions that I'm going to be following are available on the CFDE training website, which I'm going to share the link here. So I say broadly, I'm broadly going to be following those changes. There is some steps in the middle where I'm going to be taking a slightly different approach, and I'll make that very clear when I get to those steps. But if you are curious to follow along after the course, that's a resource tutorial about using kids' first data on Cavatica to do an RNA sequencing analysis. All right, so let's get started. So I think that I'm going to get started by going to Cavatica, which is cavatica.sbgenomics.com. And I'm going to log in. That's where I'm navigating to start. And so I am set up with my ERA Commons ID, so I'm going to log in with that. And it's going to ask me to authorize, which I do. And I'm going to reload the page. And there it goes. Okay. So if you're following along and this is your first time on Cavatica, your page will look a little different than mine. So mine has a lot of my projects that I'm a part of. It has an overview of some of my analyses that I've been working on recently. I'll come back to this again at the end. I'll kind of bring us back to this homepage to review. But if you, like I said, if you don't have any projects yet, it'll have a different view and it'll invite you to create your own project. And so you can do that by clicking this blue button down here that says Create Project with a plus sign. And so this is where we're going to set up our first project. So for our name, I'm going to call this Power Up August Demo. And that will be my project name. And then it asked me to choose a couple settings. So for the billing group, so if this is what we were talking about on Monday, that if you email the support or you use the question mark at the bottom, they can set you up with a pilot funds billing group, which can, all the costs for the analysis will go to that group. And so the pilot funds is $100. Our analysis today, I ran it last night, it cost me 8 cents. So you can imagine that if you have $100 and you're spending 8 cents at a time, you can do quite a bit of RNA-seq, just on the free pilot funds alone. So I'm going to choose a billing group that I have set up for this course as part of my training, and I'm going to use that for the cost today. The location, I leave the default, so we're going to use AWS computers as part of our, so like I said, this concept of cloud computing is we're borrowing other people's computers. And so we're going to borrow AWS's computers today to do our analysis, to run the computers. We're going to leave spot instances on. What that means is that, in brief, it means that our run might take a little bit longer, but the counter is that our run is a lot cheaper. And I'm generally going to choose that box because I like to save money. But depending on, the money versus time, I'll leave that to your own decisions. And then under advanced settings, I'm going to choose one additional setting. I'm going to allow network access. So what this does is when we are a little bit later today, we're going to be looking at trying to bring GTEx data into our project. So we need to allow network access to be able to find that file and pull it into our project. And so this will allow us to be able to do that later. And you can change this down the road, but I'm going to set this here while we're setting it up. And I'm going to create my project. So once you do that, it takes you to this homepage. So Cavatica is kind of organized around this concept of projects. And so everything within a project is kind of organized neatly. And then you have specific files and analyses that are associated with that project, as well as specific folks and people. So one of the nice things about Cavatica is that you can invite people to join. So for example, and I'm not exactly sure what Sangeeta's username is, there she is. So if I want to work with Sangeeta in my project, I can invite her to join. And then if I click refresh, there she is. And so it makes it really easy to add collaborators. This is especially really powerful if you have collaborators at different institutions. So we were talking a bit about, if I'm at Harvard and my collaborators at Hopkins, well, the computers, I can't get into their system and they can't get into our system. And so it's really easy. I just add Sangeeta here, and now she has the same view into the files and analysis. It makes it really easy to share and collaborate together. This big table on the left-hand side, this is just you can imagine like a whiteboard. It gives you some information about how to get started, but you can also turn it into a whiteboard. You can write hello world. And so just make notes to yourself about what this project is, future analysis ideas, things like that. So it's just kind of a space to take some notes. All right. So to do any work in here, though, we need some files. And so you can see that, files are the basis of every analysis, but we don't have any files yet. So let's find some files. And so to do that today, so there's a couple different ways you can add files, which I'll kind of briefly show up here. And so Kavatica has some public files that are available. These are a lot of reference files. In fact, we're going to pull a public file or a file from a public project in a little bit later. That's where we're going to get one of our reference files from. You can upload files from your computer. You can FTP a file on the internet. If you have what's called a DIRS manifest of files from another NIH program, you can pull files that way. We're going to show that a little bit later as one of our methods for pulling GTEx data. And if you have your own volume, your own AWS account with your own data, you can plug it in as well. And so Kavatica will be able to touch that space and be able to interact and bring those files into analysis. So you can kind of connect any data volumes you have as well. It allows you to be able to work in this kind of user interface space. So not listed here is that we can actually also push files from the Kids First portal. And so that's what we're going to work on today. So to do that, I'm going to navigate to the Kids First portal, portal.kidsfirstdrc.org. And when you navigate to the Kids First portal it will ask you to log in. And so it provides an overview of this is the amount of data, and these are the data files that are available. So we're going to log in. I'm going to log in with my ERA Commons ID again. But anyone can create an account with a Google authenticated email address if you don't have an ERA Commons. So again, if this is your first time navigating to the Kids First portal, it might look a little bit different than mine does. It also, the windows, the tabs will kind of readjust depending on how wide your screen is. So things might not look exactly the same as mine. But I'll show some highlights here. We have this up top is my authorized studies. So these are the controlled access data that I have available to me. And I'll show you how you can kind of connect your account to be able to access those studies that you might have received access to through dbGaP. Likewise, over here, this is my Kavatica projects. And so you can see there's my project I just created, Power Up August Demo, with 2 members. That's me and Sangeeta. So it's already recognizing and showing that that's connected. If you are logging in for the first time and haven't, it won't be automatically connected. And so I'll show what it looks like. It might look like this: to analyze Kids First data in the cloud, connect to Kavatica, and it gives you this button that lets you connect. So when you click that button, it might ask you to log in again or re-authenticate, but you can see now I'm plugged back in. And so it connects these projects that exist over in the Kavatica space to the Kids First space. And so that's necessary because what we're going to be working on now is finding files on the portal and pushing them over to Kavatica and making them visible. Is anyone getting a connection error with Kids First and Kavatica? I am not, but I'm not sure if anyone else is. You could try maybe refreshing your screen. I'm not sure, but I'm gonna keep moving forward and hope that maybe a refresh will unlock. All right, so other features on the portal, we'll stop in on the studies view very quickly. So the studies view provides an overview of the different Kids First studies. And so I'll highlight, because the question was yesterday, it's about controlled access data. How do we find it? How do we apply for it? So all of our all the data on the Kids First study is linked through a dbGaP release with these IDs, this PHS, and then a set of digits. And so if you're interested in a particular study, if you're interested in Cornelia de Lange syndrome, you can click on this link here, this PHS number, and it takes you to the dbGaP website for the Cornelia de Lange study, provides information about Cornelia de Lange syndrome, what this is, It tells you about the consent of the participants and provides some information about the study. And then at the top, there's an opportunity to request access. So this takes you to a page where you can log in and then submit an application for a specific research project if you're interested in a particular study. And you can combine, so if you're interested in multiple studies, so if you're interested in combining Cornelia de Lange with the hernia study, you can submit, both of these projects or request access on one single request. So you could include both of those IDs as part of your request. So you don't need to make two individual requests. You can just make one, and they can provide access to you on both. From there, it's up to, so the Kids First DRC is not responsible for reviewing or approving access requests. That's part of the Kids First Data Access Committee, which is an NIH group. But I understand they move relatively quickly. I think within 2 to 3 business days, they usually have turned it around and provide folks access. So that is a way to carry on. All right. So this is today specifically, I haven't really talked about our specific example that we're going to be looking at. So today we're going to be looking at a set of data all the way down here at the bottom. Our example we're going to be looking at is some data from children with T-cell acute lymphoblastic leukemia, or TALL. And so for our Kids First TALL study, we have a collection of both genomic data and transcriptome data, which is good because I'm choosing to use open access data for our demo. And so this is why I'm choosing to do a transcriptomics analysis and RNA-seq analysis. And so this is what we're going to be looking at. And so TALL is a type of leukemia. And one of the things about TALL is that it has a higher incidence in boys than it does in girls. So boys are more likely to be diagnosed with TALL than girls are. So today, as a demo, we're going to look into that a little bit, and we're going to compare gene expression in leukocytes from boys with TALL versus girls with TALL. And so that's going to be our analysis. So they're going to be our two groups that we're going to be comparing expression across. All right. So to find that data, we need to go into data exploration. And I've got my query saved, so it'll save your query for you if you come back. But I'm going to clear this out, and we'll recreate this here this morning. And to start from scratch. We'll let everything load up. So what we're going to be doing is so the analogy with this one, like I said, it's a bit like the library where you have thousands of books or thousands of articles, and then you're applying filters, based on the author or the journal title, to narrow it down to a specific set that you're interested in. So we're going to be doing the same thing with the Kids First data, is that we are going to let it finish loading. And then once it does, then we're going to be applying these filters in order to be able to find the set of participants that we're interested in. But what we would be doing, and I guess what I can start working on showing, is that we can apply specific filters. So we're interested in a number of participants. Here's our 34,000 participants in the Kids First data set. So we want to narrow this down. We don't want all 34,000. So why don't we start with, we want to do RNA-seq. So let's just look at RNA-seq. So we can do that by applying a filter over here on the left-hand side for data file. And then under experimental strategy, we can apply a filter for RNA-seq. Apply. So we're going to apply that. And so you can see now I've got this filter up here, and now all my windows are refreshing, and you can see we've narrowed down our data quite a bit. So not every sample in Kids First has RNA, and so we've shrunk down to now we're only looking at 2,800 participants. And again, so we can look across. If we scroll down different studies, you can see we have a lot of RNA from the TALL study, which is good. That's where we're going. But you can also see other studies that also have RNA available. So we can apply a filter for TALL. You can actually click this pie piece in this pie chart, and that will apply a filter. But I'll show you can also get there if you go to the study window. and then do study code. And we're going to choose TALL and apply. All right, so now we're getting smaller and smaller. So now we've got 1,000 participants here, and we've got 9,000 data files, which is still a little unwieldy. So let's continue to narrow it down a little further. So if we click on this data files tab, this shows you the 9,000 files that are available. And it has a couple different data types. And so we have these raw gene fusions. So this would be another reason why you might be interested in doing RNA-seq if you're looking at gene fusion calls about large-scale chromosome rearrangements. RNA-seq is very good for that. But we're going to be interested in gene expression quantifications. And so we're going to narrow that filter by data type. And we're going to choose gene expression quantification. So that's under data files, data type, gene expression. Apply. All right, so now we're down to 2,000 files. So we're honing in here on a smaller and smaller set. We can narrow by file. So you see our file format now is all these .tsv files. And so these are going to be TSV is tab separated values. So it's a big table where, a column where all of our rows are going to be genes, and then we're going to have expression values in our columns across the table. And so then we're going to apply one last filter. So this is a large number of samples, and we could run it on this many samples, but for the sake of our demo today and making sure that we can run it in the time allotted to us, I'm going to apply another filter to narrow down this number of samples. So I'm going to do that by just selecting, narrowing our number of participants to a smaller set. So I'm going to apply a filter for age at diagnosis, and I'm going to set age at diagnosis to be 1,000, less than 1,000 days. So these are children who are about 3 years old or younger. Apply. And there's not, I'll be clear and upfront that there's not a biological reason I'm necessarily choosing to do this. It's mostly for the sake of the demonstration today to allow it to be viewed. Heather asked, those CSVs came from the Kids First analysis pipeline for gene expression quantifications. Great question, Heather. Yes. So where do these files come from? How do these come to exist? So that's a great question. And so these came from our Kids First analysis workflow, which I can show in two spaces. And actually, give me 2 seconds. I'm going to pin that, and I'm going to come back to that. But that is something I do definitely want to talk about. So we're going to take these 122 files. That's a pretty reasonable amount to work with. I'm going to select all of them, all 122, and I'm going to analyze them in Cavatica. Click this button. And so now it says you can copy these files. So I'm going to choose to copy them to my Power Up August demo. I'm authorized to copy all 122 because these are all open access files. And I'm authorized to copy them. Great. So I'm going to do it. I'm going to click the copy button. And so as it's working, I will say a caveat is that the button says copy files. We're not truly copying files. What we're doing is we're copying a shortcut in Cavatica. The files only exist in one, cloud workspace, and that is what allows us to be able to copy those files. All right.
I'm sorry. Could you demonstrate how to copy the files again, please?
Sure. Yeah. So I'll clear my selection. So I'm on the Data Explorer. And I'm on the Data Files tab where it says this many files. And then I'm going to select all of the files. And then so I've got 100 items selected. And then I'm going to choose select. So what this does is this is paginated to just have 100 at a time. But there's actually more per view. So I'm going to add an additional. So I want to select not just the first 100. I'm going to select all the results. And so now I get 122. And then from this point, I'm going to click the Analyze in Cavatica button. And then that opens this, and then you can choose which project. So that project we created earlier should be present in here. And then you click the Copy Files button. and then that is going to send a copy of those files. Yeah, so if you're seeing just 100, make sure you're choosing that additional step here where we're selecting all results. And so that's how we go from 100 to 122. All right. So I'm going to take two more things before I move forward, before we leave the portal. I'm going to go back to the biospec. So one person asked yesterday, is there a way to get, or maybe this is Monday, is there a way to get data out of the Kids First portal and save this as tables? And so the answer is yes. And so I'm going to go back to my biospecimens. And especially important to me is that I want this external participant and external sample ID because I'm going to use those as part of a join that I'm going to do a little later to patch some clinical data. So I'm going to select my 61 information about these 61 biospecimens, and I'm going to click this export as TSV. That's going to download a small file to my desktop. And then I'm going to choose, go back here to this participants tab, which if I scroll over, oops, also has that external participant ID. And this has our sex value because that's what we're going to be filtering and sorting these participants on. That's our, our value we're trying to do the RNA-seq on. So I'm going to likewise save these, and we're going to download this as a TSV as well. So we're going to have those on our desktop, and we're going to come back and use those in a little bit. Okay, and you can choose additional columns using this gear. So you can make specific columns appear and disappear, and you can actually reorder the columns as well. So if your columns don't look exactly like mine, and then you can reset the default at the bottom as well. Okay, so it said we copied the files. So now we've got to go back to Cavatica, and if it's been too long, then Cavatica will log you out as a security feature, but we can log back in. And ta-da! So now if I go back to my dashboard just to refresh, so here's my dashboard with me and Sangita and Hello World. And then if I click on Files, now you'll see that we have files to work with. So that's great. And so you can see from the output though, so we have these files here, and we've got some that say .callisto and some that say .rsem. And so what does all this mean? So this is where I'm going to take that pin out of that question related to where did these files come from? How are they created? So all of our files in the Kids First portal are generated from our Kids First workflows, which you can find on Cavatica and you can also find on GitHub. So these specific files came from a tool. And if we go into the public apps section, so we make all the apps available. So if you want to bring your own data and run the Kids First RNA workflow on your own data, you could do that as well. Now I'm going to search for RNA, and I'm going to find the Kids First DRC RNA workflow. And so what this is, so this was developed by us, the Kids First DRC here at the Children's Hospital of Philadelphia. And it includes, so it's got a great README here, which talks about the different tools. And so it allows you to be able to trim adapters from raw reads. Then it runs that alignment using the tool called STAR. And then we've got two different quantification tools. We've got RSEM and we've got Callisto. And then we've got a couple fusion callers as well, StarFuse and Ariba. And we also have added an additional tool for detecting alternative splicing, RMATs, in order to be able to assist with our data here. So the reason why we have two sets of files is that we're part of our software runs it through two different quantifier tools. So we have this RSM tool and the Callisto tool. And if you're curious to read more, we link out to those, the docs and the publications about those tools. If you want to compare the differences between them, why you might choose one or the other for your analysis. So I'm going to go, so this is it if you wanted to read a little bit deeper about the tool. And then it's also available on GitHub as well, the same tool, and I believe it probably links. Let's see here. No Barkovac public app. Okay, or we can link to the GitHub release. There it goes. So then you can find information about the code in our GitHub release. Okay. We're gonna go back out though. So this is where these files came from. So each sample has two files. It has one RSEM file and one Callisto file. So for today's demo, we're just going to look at the Callisto files, which means we don't need the RSEM files. So we're actually going to delete those out of our project as step one. So if I go back to my project and I go back to the Power Up August demo and I go back to my files, and so you can see this, Callisto and RSEM. So we want to keep Callisto and want to delete RSEM. So we can do that by searching. So if we search for a string, rsem.genes.results.gz, and hit enter, then what the platform will do is it will pull out just those files that have that particular string. So while we're waiting for that, let's go ahead and I guess we can start to set up our app. And then we might have to come back and pick up our files in a little bit. So if I go to, so we'll come back here. So I'm going to go to our apps for a second and we'll revisit. So each specific project has apps that are loaded up in them. As well as, but you can copy apps from the public, kind of public app store, like you imagine on your phone. But the public apps are all, free to use on Convatica. You don't have to, pay to use the app. You might be paying to run the app in terms of running it, but there's no charge, to buy, access to DE-Seq2 or anything. Everybody can use the app itself. And so we're going to add an app to our project from the public apps. And we're going to add an app called DE-Seq2. And so there's DE-Seq2, performs differential gene expression analysis across 2 or more study conditions. That sounds great. That's what we're going to work on today. So we're going to copy that to our project. It says the app has been copied. So if we go back to our project, for our Power Up August demo, under the Apps tab, under DE-Seq2, and so now we can see we have an option here. We can click it. And so just like the Kids First workflow, it also has a README here about the tool. It provides information about the inputs that we're going to need to run the tool, and it provides a lot of description and details. And so again, this is great to read if you need help troubleshooting. So we are going to run this app. And what we're doing now is when we click that run button, it creates what's called a task. So we've been slowly working our way over the left-hand column up here. So we had our dashboard, then we looked at specific files, then we looked at our apps, and now we're up to tasks. So a task is a specific running of an app. And so we're going to run DE-Seq2, and this is when I click that button, on August 21st at this time UTC. And so this is what is going to be, and I could rename this to something more intuitive, or I could keep the timestamp if that makes the most sense to me. And so the way the app works is that it has certain inputs. So we want to input specific information into the app. Then we choose specific variables that we want to look at more in depth. And then after we run it, it will provide the output over here. And so you can see the input, on the input side, we need to add expression data files. We need to add our gene annotation layer. So this tells us what the genes are in the genome file. And then we need a phenotype data file. This describes, this is what we're going to use to tell the software, which samples come from boys and which samples come from girls, because that's going to be our variant of interest that we're going to apply. Okay, so we need some files. So I'm going to hopefully have stalled long enough to allow the files tab to load for me. There we go. All right, so we're going to try again to get rid of those RSEM files. So we're going to click RSEM, and we're going to search. And so what this should do is it should eliminate those, it should pull out just those rsem.genes files for us. We need 3 things here for our DE-Seq2 run. We need the expression data, which we got. We do need to patch some metadata fields for it in order to be able to work. We need a gene annotation file, and we need some phenotype data, which we're going to create. So if I choose this gene annotation, and it even has some little question marks here, which tell you a little bit more about what these particular, is expected here. So for gene annotation, we need this GTF file. What this does is it tells gene names and gene IDs, and this is going to be important for for figuring out, what genes are actually overexpressed and underexpressed and applying that information. So we can find this gene annotation file by looking in the public projects. And then Kids First makes available all of our reference file in this public projects called KF-references. And then we go to files. And so this is all of our references that we use. So when we run those workflows, like the Kids First RNA-seq workflow, we pull files out of this project. And so if you're interested in, working with our data, we recommend using this same project as well. And so we have links to, it's part of that description, that README of the RNA-seq file. We have recommended inputs, which link to this page, and that kind of is another way to direct people here. So we're going to search for, and hopefully search works for me. We're going to look for our GTF file. Here we go. And we're going to look for GenCode V39 primary assembly .annotation GTF. And so this is our reference file that we're looking for. This is a .gtf, so it has a specific format where we're mapping out the locations of the genes in the genome. So this is the file we want for our analysis. And so I'm going to copy this. I might even copy the name of it. If I can, we'll just copy it to our August demo project. And we're going to copy it right there. And so it says it's copying right now. And we copied. And so if we go back to our file, I'll even click on this just to compare. And I'll share this with folks in the chat in case folks are curious about the exact name of the file we're looking for. But now if I go back to my demo project and I go to files, boy, we're struggling. There we go. So now we can see we've got this GTF file in here as well. All right, so I'm going to try one more trick here. So I'm going to sort by extension, and I'm going to just choose we're going to try again to get rid of these RSEM files, which are the results, and we're going to apply. There we go. That worked way quicker. So these are all of our RSEM files. So there's 61 files here, and we're not going to use these for our analysis today. So I'm just going to select them all, and I'm going to delete them out of my project. And it's just going to kind of tidy up our project a little bit better and make it easier for us to work with. So since the delete is finished, you can go back and click refresh. So now it says there are no files, but it's because we still have that filter applied. So if we clear our selection, ta-da, all of our files are back. So we're safe and sound. So now we've got our files here. And so we have to actually patch some metadata in order to be able to get our workflow to run properly. And so if we click on this first Callisto file, you can see that when we copied the files from the Kids First portal into Cavatica, some of what's called the metadata, this is kind of information about the file related to who this sample came from, didn't copy over properly. So this is an error from a recent update to the Kids First portal, and we've got the data team working. And so this is where we're going to be kind of patching this to be able to get it in a state that will allow us to be able to run the workflow. How do we get the GenCode file? So that's under public projects, and then it's under Kids First references, and then it's in the files tab. And so I'll share this. And then it is specifically, if you search based on extension, GTF, apply, it's version 39 primary assembly. I can even share this specific link, but it is helpful to see how I got there. Okay, so we need to patch the metadata. And so this is what we're going to work on now. So if I go back to my files, and we're getting ready to step out of Cavatica here, but I need one more thing out of this project. So I hope it lets me in. We're going to go, we're in the harmonization projects. So this is where it's tricky. You got to see, which project you're in. We're in the references. We need to go back to our demo. We're going to go back to the demo. We're in the right spot. Go to the files. We're in the right spot. So what we're going to do is we're going to select all of our Callisto expression files, so our 61 files. And what we're going to do is we're going to generate a metadata manifest in CSV format. And so what this is doing is it's taking all of those metadata fields that we showed for that first file, and it's making a big table of them. And then it's going to save it as a manifest that we can then download. So we're going to download this small file and bring it to our desktop where we're going to do some editing in Excel. So if I hit refresh, then we should see our new manifest file.csv. Perfect. So then I'm going to check this box, and then I'm going to download this to my computer. And now it'll let me download that file. And so, you know, the perk we like to say, oh, you're working in the cloud, you don't need to download files. Every once in a while, you know, it's easier to download the file. And you can see that instead of downloading 61 files, all of them are almost 3 megabytes, you know, we're downloading this one small manifest file that's 57K. And so it's, you know, much easier, quicker data transference instead of trying to copy, you know, for my small example here, 61, but you can imagine scaling up, that would be prohibitive where you'd be able to download all of these files themselves. All right, so now we're going to step out of our tab here, and we're going to step into a tool that many of you are familiar with. We're going to look at some Microsoft Excel. So I'm going to pull up on my desktop. So I'm going to put Cavatica away for a moment. We will return. But I'm going to put up on my desktop 3 files. So there is the manifest that I downloaded out of Cavatica. And then these are the 2 files we downloaded out of the Kids First portal, the participants and the biospecimens. And so this manifest from Cavatica, I'm going to open this with Microsoft Excel. And so this is just a big table. And so it has information about, so it has this specific ID, and then the name of that file. And then it has information about, you know, the study and the diagnosis of the participant and the age when they were diagnosed, and things like that. But we actually need kind of 2 additional fields in here. We need a sample ID and we need the sex of the individual in order to be able to run our RNA-seq analysis. So for the sake of making things a little bit easier, I'm actually going to delete a fair number of rows out of here. So I'm going to delete nearly all of these except for this external sample ID, because we're going to use that to do some joins. And I'm going to keep the external participant ID as well. Delete. Delete. So for the sake of our example today, we're going to keep it very simple. But you can include a lot of those extra fields to do controls for your analysis in order to be able to set things up.
So some questions about the public projects. Where are the Kids First references? So thanks, Sangeeta. And I'll show that one more time, but I do want to keep moving. When we go to add that file, I'll show again where I got it from. I do want to keep going with my table here. So let me share my screen once again. So let's see here. That is, so we're back in Microsoft Excel. So I'll show kind of why am I doing what I'm about to do? Why do we need to set this up this way? And how do I know that we need to set it up this way? So let me pull up, if we go back to our browser window, we go back to our apps, and we learn about DESeq2, and we get this README for DESeq2. One of the instructions is that we need the CSV phenotype data file, which includes for each row with sample ID in the first column. And these sample IDs need to match those in the input file's metadata. Also, a single line header with the variable name should be included, and it gives an example. So this is why we're having to go and set up this file. So this is kind of, and it gives you directions about how to do it. So why am I doing this, and how would I know to do this? So again, it's in the directions of the workflow. So I just wanted to provide that context. Okay. So we need to make a new file. So here's our new file, and then in our first column, we need something that's a row that says sample ID. So I can add to the left here. I can right click and insert. And we can add a new column that says sample ID. And so now we need to fill these in. So the sample IDs are the format BS underscore. And so this is where we have to do some kind of Excel tricks to pull in some additional information here because I don't see that represented in my table. Whoops, we definitely don't want to do that. Okay. So to do that, we're going to do an XLOOKUP. So I'm going to add a tab, and now I'm going to bring in that data, one of our files from the Kids First portal. I'm going to bring in that biospecimen data. And so we're going to open this file with Microsoft Excel as well. And so what we can do is I'm just going to copy and paste into my manifest file, just so that way I'm working in one space. And so you can see what we have here. This is our sample IDs in BS underscore format. And it has these external participant and external sample IDs that I can do this join on. And so this is kind of you can imagine we can do a lookup where, okay, I would start with, 0021, and then I can scroll through this sheet and find 0021 and then copy this over for all 61 participants. And that would take a really long time. So we're not going to do it that way. We're going to do an XLOOKUP, which is an Excel command where you do equals X lookup. And so we're going to look up a specific value. So we're going to join on this sample ID. So we're going to look for this. Basically, what we're doing is we're searching for this number 21. And then we're going to look up where are we going to look for it. We're going to look for it in this column. So we're going to look for 21 in that column, comma. And then we're going to return this value in this column. So if it finds the second value in the second row, then it's going to return this in the second row. If it finds the value in the 20th row, then it's going to return this in the 20th row. Close parentheses, return. And so you can see it pulled that value for us automatically. We can then drag and drop this all the way down our column. And we matched up all 61 of our samples just like that. So now we are getting closer. So we have our phenotypes with our sample ID in the first column. That's good. But what's not represented here is our biological variable we're interested, which is biological sex of these individuals. So we're going to add another column right here. And we're going to add, we'll call it sex. And now we need to do another lookup. And so for the sex is in the participant because sex describes a participant, doesn't describe a biospecimen. So we're going to open our other file we downloaded from the Kids First portal. And so this has a link where we can join external participant ID with sex of the individual. So those are our two columns we're going to look for. So I'm going to go back to my manifest. I'm going to add another sheet. And we can even label these just so that way it's more clear. These are biospecimen. And this is participant. And I'm going to copy this participant table. And so we're going to do another XLOOKUP in this sex column equals XLOOKUP. We're going to look up this value, comma. Where are we going to look for it? We're going to look for it in the participant ID column, comma. We're going to return the sex of the individual from the sex column, close parentheses. So there's our sex of that first participant. Scroll all the way down. And there we go. So there's the sex of all of our participants. So now we've got this file we're going to use for two different things. So one, we're going to feed this into DESeq2. This is going to be our phenotypes file. And then two, we're going to use this file to patch the metadata in Cavatica as well. So we can label all of our samples as being male or female. So I'm going to do file save as, and I'm going to call this phenotypes.csv. We won't call it .csv because that will be saved. We're going to save it to our desktop. Save. And so it's saying that we can't, because it contains multiple sheets, we're just going to save this sheet that we're on. That's okay. That's what we want. Click OK. And so now we've got a new phenotypes file. Yes, Sangeeta, I see you have a hand raised.
Yes, I just wanted to add just a little bit. Whenever I've used VLOOKUP or HLOOKUP, I have needed to make sure that the column that I use for looking up the value, it needs to be sorted. So I just wanted to make sure if this file is already sorted, or the column was already sorted, or did you have to do that, or if the users were to use a similar file later, do they have to make sure that they have sorted the columns?
I have not sorted, so I get it from the Kids First portal. I think so. The default in the Kids First portal, it's sorted, looks like alphanumerically by participant ID. And so, but we can do kind of a confirmation we can check. And so we can say, for example, just pick one at random, this participant. So this participant, if we look them up in the table, they should be female. And so if we go to participant and look for them, then it says that they are female, so that is true. And then likewise, we can look up their biospecimen. And so it should be 9 NYJR. And so if we search for that, and hit enter, then their biospecimen is 9 NYJR. So just kind of a confirmation. So it is good to double check, make sure that these joins, we're not joining the wrong thing, because that can cause some trouble downstream. Can you share the file here? So I'm going to upload the file to the project. I don't know if I can share a file through a Zoom chat, but I can share through the project at the conclusion of the demo. Okay, so we got our phenotypes.csv. So this is good. This is what we're going to need to run our software tool. So we can now close Excel, just minimize Excel. Bye-bye for now. We will come back later. Okay, so now we're going to go back into our browser window. So we're back in the cloud. And we're going to go back to our projects and back to our demo project. And so we're going to go to our files. And so we're going to select, again, not our manifest and not our GTF. We're just going to select these 61 expression files. And over here on the right-hand side, it's hidden a little bit. There's this ... We're going to click that ... and we're going to click Edit Metadata with a Manifest. And so this says you can upload a CSV file, which contains these DIRS IDs, which is that second column, which can then be applied to the file. So we can browse from the computer, or you can drag and drop. So I'm going to drag this phenotypes file that I generated, and it's complete. And it says, what metadata do you want to add? We want to add sample ID, and we want to add sex, because that's what we're going to be looking at. Submit. So now it is submitted for processing. And so we'll let that go. So if there's this little bell up here, which tells you it's edited and that it's complete. And so if we click on this top sample, we should be able to see that sample ID is this BS. So we were able to patch it successfully. And then if we scroll down to the additional fields, we should have an additional field for sex. That corresponds to the sex of the participant. So that's great. And then we can actually go back. So if we go back to our big file list, we can view this as well in our column. So you can see here all of those sample IDs. So if I go, you can adjust these columns. So sample ID is shown, but I can also scroll down and add a column for sex. And so you can see there's our samples with the sample ID and the sex of the individual. Great. Okay, good. So we are just about ready to put all the pieces together now. So we have all of our pieces. We have our expression abundance files. We have our GTF file, and we have our phenotypes file. So those are the three inputs we need to run the workflow. So let's go ahead and plug it in. So I'm going to go back to my task. And we're going to go to DESeq to run. And for expression data, so now this is saying, it tells us what types of things we're looking for. So you can select files. So we're going to select these TSV .gz, which is a compression format. And we're going to select all 61 of those, and we're going to save it. So now it's basically saying for this expression data input, we want these 61 files as our input. For the gene annotation, again, this is our GTF file. And once I click run, I'll show again where I'm pulling this file from. So it recommends extension GTF, and we only have one of those in our project. So we can save that. And then phenotype data, it provides information about what this is, a CSV file with the sample ID in the first column. So we can select that, phenotypes, save. All right, so now we have a chance. So that's kind of our required inputs. In the middle column here, this is the settings that we can use as part of our, the settings for the software tool. So we can give it a name. We'll call it Power Up Aug Demo. Or maybe we'll call it, and then covariate of interest. So this is required because this is how we're going to, sort these files. And so we want our covariate of interest to be the sex of the individual. So we're going to type sex there. And then we have one more required field, which is which quantification tool was used to generate these. And so these were built by Callisto. So we're going to choose Callisto. So now we've set everything up. And so we should be good to go. And so we're going to click the run button. And away it goes. So what happens when you click that button? So what happens now is at the very beginning, and we're in what's called a queue. And so we're waiting to be, like I said, you can imagine like if you're at the grocery store and you take a ticket waiting to be helped. So we have our project and we are waiting in line to borrow, this cloud computer to be able to do it. And so you can see we've already jumped forward to the next step. So some of the instances for this task are being initialized. What that means is that we've been connected with a computer and they say, yep, you can borrow this computer for your analysis. And we're starting to turn the computers on and spin them up and get them going. So this is going to take another minute or two, but everything is looking good so far. So I'll show one more time while we're waiting here. Like I said, last night when I ran this, it took about 8 minutes. So while we're waiting, I can show, I'll show one more time where you get this GTF file from. From any page, from the home page, you want to click on Public Projects. And we can navigate away from that task page because it'll be running in the background. So we don't need to keep that window open. And then on the task page, on the Public Projects page, you scroll down, there's a project called KF References. We're going to click that. And so this tells us this project houses the reference files that we use for KF Public Apps. And then you want to click on Files. And then we want extension .gtf apply. And then we've got a couple different versions. We've got some older versions, but right now we're on the version 39 primary assembly. Now I click that and then click copy it to my project. So that's how I did that one last time. Okay, so let's go back to our dashboard and see how our analysis is going. So it's still running. So if we click on this, it says some of the instances are being initialized. So that's good. So like I showed, you don't have to stay on this page the entire time. It's running, in the cloud. So it's not running on your laptop. It's not running in this browser window. It's running on a computer that's managed by AWS. So you don't need to keep this window open. You don't need to, wait for it. You can go to lunch, you can take a break, depending on how long you expect the analysis to run. But I don't expect this one to run very long, so I think we can hang out for a moment. But I will show some additional features on this page. So while it's running, there's a button here. We could click the abort button. We're not going to click that one, but you can click View Stats and Logs, which tells you a little bit more about what's happening under the hood, so to speak. So we click this View Stats and Logs button. It tells us we just started. We don't have any stats yet. be more patient, David, is what it's telling us there. So we don't have any stats to read yet, but as it runs, this stats page will populate with some information about, the run and if there's any errors and things like that. Do we use the same reference files for all Kids First genomic data sets? So that's recommended. So especially if you want to use, if you're trying to use Kids First data, or if you're trying to compare Kids First, your own data to Kids First, and so you want to kind of harmonize, then it would be recommended that we use the same references. So I'll highlight really quickly that we've jumped forward in progress. So now we're not initializing, now we're running. It's doing the computation right now as we're talking. And I'll show one more time, like, how do we know, like, where do you know to get Gen code v39 primary assembly? Like, David knows that, but how do you know that? So that's in those public apps again. And I'll show if I pull up the Kids First DRC workflow for RNA-seq. And so you see we have a couple different workflows. We have like alignment and haplotype caller and structural variants, but we're interested today in our RNA-seq. And so as part of the README, I talked about this part with the different software tools, but if we scroll down, it gives you more specific information about the input and all the settings that we choose and recommend. But if you keep scrolling down, we have suggested references down here, as well as in the inputs. And so this would be where you would get that. Here we go. Suggested reference inputs are. And so this is where, well, what reference FASTA do we want to use? And it gives you this link. Well, what annotations do we use? And it gives you this link. And so it gives you specific information about, what types of references and that kind of thing. So this would be where you would find that information on your own. All right, so let's go back to our project. It's still running. So we're at about 5 minutes. And it's still running, but that's okay. Other things that I can share while we're waiting for it to run. So I highlighted this button down here, this question mark, yesterday as a way to ask for access to the pilot funds. This is also a great place if you're stuck, if you need help, if your workflow doesn't work. What it does is it kind of records, where you are and what workflow is, and then you can write, I need help. My workflow doesn't work, you would obviously provide more details. And then you can click this send button, and that will send this task to a Valsera support person. So they'll be able to review it and then try to help you identify what any issues are, and try to help you do any troubleshoot to get everything working. So it's kind of hidden down here, this little blue question mark, but it's a useful tool as well. All right, we're at 6 minutes, so that's promising. 7 minutes, so I just feel like we're at New Year's Eve waiting for the ball to drop. But let's see, what other things can I talk about to fill the time with? I guess I can talk about where we're going to go next. So right now we're running a task, which is a bit like submitting a job. So we've packaged all these things up. We packaged all these files, and we packaged our annotations, and we packaged our phenotype description, and we kind of said we're going to run it in this software, and then we just kind of put it up and ask the computer to run this computation. You can imagine, like, if we had some really complicated command in our command line, and then we hit enter. Now the computer is thinking about that that one entry. Another way that we can interact with our data is what's called a kind of an interactive, where we can really, kind of use commands, much smaller commands, kind of less computationally taxing, in order to be able to work with our data more rapidly. And so we can do that in what's called the Data Studio, which is where we're going to go next, once our analysis wraps up here. Where we can actually dive in and look at the data a little bit deeper. So our task is still running. Give it another minute or two. And I guess I can also highlight while we're waiting for this to run. We are going to be pulling some data from GTEGs. So maybe I can start getting that set up while we're waiting here. So we can add another window. So we're going to go to our GTEX portal. So here's our, so now we're on the GTEX portal. And so we're interested in bulk tissue expression. And we want gene TPMs. And so we can choose, so remember GTEX is based on particular tissues throughout the body. And so for our leukocyte example, we could compare to our EBV lymphocytes might be, an interesting kind of control that we could compare to. So maybe we're interested in this file, and I'll show how we can pull that file into our project here in a moment. So we're going to come back to that, but we'll keep that bookmarked on another. Let's see. And it's completed. So if we go back to our dashboard, you can see that our task is completed and it has green. That's good. So if we click on the task, it took 9 minutes today, and it cost us 5 cents to run this analysis. And it gives us some additional information here. These are all the output files that it generated as part of our analysis. All right, so let's see what we learned. And so DESeq outputs a couple different things. So it outputs this nice summary report in an HTML format. It also gives you the raw data in these text formats, and then it makes this big R data package, which we can explore. So we'll first look at the summary report, because it's kind of a quick and an overview. And so it tells us what we inputted, and so here's all of our samples and the sex of those individuals. So there's our phenotypes file that we fed in, and then it provides a PCA plot. And so from brief looking, I don't see very strong definition in our PCA plot. I don't think we're going to be able to publish this one in a really high quality journal. It seems like sex is not a major driver here, at least in gene expression between these different samples. We can look down, so it provides some overviews of these different data counts. Provide some additional plots showing you about kind of these broad level. And then one thing I'll highlight is this heat map down here, which provides information about the most strongly differentially expressed genes based on this value for sex. And so we can kind of confirm some existing things that we know about these participants. Remember, we're using biological sex as our variable here. So we've got female on the left and we've got male on the right. And so we see, for example, X inactivation is much higherly expressed in females than it is in males. So that attracts with our biological understanding. And then likewise, we have a collection of genes that are present on the Y chromosome down here. So these are also much, way highly expressed in males and virtually absent from females. So that also kind of correlates as well. So it gives us kind of a sanity check, if you will, that, yes, these samples, seem to be, corresponding with what we would expect based on, differences in the sex chromosomes. But then we might want to dive deeper into what these different, genes are that are also showing up on this table. And that would be kind of a part of a future analysis. All right, so that's the completion of our analysis. So I'll show kind of 2 more things here. We got about 20 minutes left in the class today. So I will show, so one of these outputs, as I talked about, is this .r data file. And so this R data file allows us to load up everything very rapidly in a session of R. And so if we wanted to, we could download this file to our computer and we could you know, spin up RStudio on our computer, and we could unpack this file and really kind of dig into what these different genes are and what these different counts are. But we don't have to do that. We can work in the Data Studio and use, again, the more powerful computational cloud computer instead of trying to run everything through the computer in our laptop. Okay, so we're going to hold on to here, and I'm going to go to the Data Studio, and we're going to create a new analysis, and we're going to call this RStudio. And where you can, so you can use either a Jupyter Lab, which is a Python-based kind of notebook environment, but we're going to work in RStudio, and we're going to use this particular type of computer. And so in the interactive node, everything is billed, it's kind of billed per time. And so we're borrowing a computer and we're paying a rate of 34 cents per hour. So it's kind of like the lights in your house. You know, you want to turn the lights out when you're not working on them. Or you're not in the room so that way you're not paying your power bill. So likewise, we're going to, you know, turn on the computer and we're going to use it, but when we're done, we're going to make sure we turn the computer off. So that way we're not, you know, continuing to be billed. And Cavatica has some additional features that help, you know, kind of keep an eye on you. So this suspend time. So if we're inactive for 30 minutes, Cavatica will automatically turn the computer off for us. So if we forget and go to lunch or go home for the night, you know, we're not going to get a huge bill the next day by leaving this suspend timer on. So we're going to click start. And we're going to let this spin up. All right. So this is going to take another couple minutes to spin up because we're again kind of we're finding the you know, being connected with the correct computer, and then it's preparing everything, and then it's going to get our environment set up. While we're waiting for this, I can we can go back. So again, we're in the cloud, so we can we don't have to stay on this page. It will continue to work in the background. And you can see we're at the initializing step. I'll just show one last set of features that I really appreciate about kind of the Cavatica platform. And so this is my task that I ran, it completed. I'll show two things. One is that it has all these logs and stats. And so if I click this box, it tells us about the tools that were run. We ran DESeq2. We can view the logs. And so if we really want to dive in, this is more helpful if you get error messages because then you're trying to troubleshoot, you know, what went wrong. It will provide the error messages in here. And so you can try to dig into, you know, any troubles that might have happened. So there's kind of additional information here provided. But then what I also really appreciate is that this task saves itself. And so it was run, you know, on August 21st, 2024 at this time, and everything is saved here. So I could come back next week, and if I want to run the same thing on different samples, I could very quickly click edit and rerun, and it pulls a new task, and it brings in all of that same information, and maybe I just want to change out a couple of my expression files. And so I can click that change selection and rerun everything with the same settings. So everything is here. What I love about this is that it makes everything really reproducible, makes everything really trackable. When you're writing up your paper at the end of your project and you're, well, what did I do? What methods did I do? What settings did I choose? You can go back. Oh, it's all saved. And so you can go back to that task, you know, that we ran on 8/21/24. And all of those settings are saved for us right here. So we know exactly what to put when we're trying to publish and write up our story. Okay, so now I'm going to go back to the Data Studio and hope that I have bought enough time. So it's running, so we can go ahead and begin to work in it. So we click the Run button. It's going to spin up and open RStudio. Hopefully. There it goes. And so once it loads, it's going to open a window. If you're familiar with RStudio, it should look very familiar to the RStudio that you might work with on your desktop computer. And so this is not our course this week. I'm not going to go in depth into, you know, how to use R, how to do analysis, but I will show some kind of basics here of how we interact, so and how we open that data file that we found. So we can go onto, so here's our R console, but we can also have a terminal. And so we can explore this directory is empty. But I actually know from experience that our files are in a directory one above us called project files. And so you can see, oh, here's all of our, all of those files in our Cavatica project. Here's all those Callisto expression counts. Here's our GTF, here's our phenotypes, here's our manifest, and then here's our output. So there's our data file. So that's what we want to open up because that's where all of our data is being stored in this nice package. So what we're going to do is we're going to go back to our R console and we're going to load some data. And so we're going to load a particular file and we're going to enter that same path, / project files. And then we're going to slash, what was the name of that file? Power up aug demo. Power up aug demo.r data. Enter. And so what this is doing is now it is pulling, it's opening that file. You can imagine like a zip file on your computer. And so we're unzipping everything and loading it up in our environment. So it's going to take a moment to kind of unpackage everything and get it all set up for us. And now you can see we have these additional data tables and variables that are already loaded up and are ready for us to analyze. So we can kind of explore these a little bit more in depth. And so it gives you kind of the list of the different genes, gives you a list of the different gene names. But I'll show we can also kind of begin to do, plotting and analysis as well. So if we install the DE-seq2 RNA R package, so under our packages, we can choose to load up DE-seq2. We can begin to kind of plot and interact with these analyses. So kind of a really simple, if I use this command, we can build a plot, kind of an expression plot, where we're looking at, from left to right, we're looking at expression of genes. So higher expression is on the right, and then fold change is shown on the y-axis. So we have higher, I believe one of these is male and one of these is female. I'd have to check exactly which way we set it up. Which one our kind of our reference and our alternate is for the sake of this example. But there's our kind of an additional graph. It kind of shows you that, we can unpack this data a little bit further, without going too far because we are starting to run out of time here. Okay, so there is our analysis in the cloud. So we have gone all the way from, the starting point to finding these data files on the portal. We moved them to Cavatica. All the way to we have a plot, showing relative gene expression according to the biological sex of the individuals. So if we wanted to take it one more step, and I can't show too much more, but I will show roughly how do we get additional data into our study here. So for example, this GTEx. What if we wanted to bring in this table for gene TPMs for cells from our lymphocytes? So we wanted to compare to some control data. So I could download this file. It would download this 15 megabit file to my computer. And then I could upload it to Cavatica using the drag and drop like I did with the phenotypes file. Instead, what I'm going to do is I can do a wget to actually pull this file directly into this interactive space. So I'm going to go to this download button. And instead of clicking it, I'm going to right click and I'm going to copy the link address. I'm going to go back to my RStudio. And so I'm going to go to the terminal. And so I want to wget that file. So I can do wget, and then I can do that URL. And so what wget is a command that lets you basically like download a file, like save a copy of this file to this location. And so that's what I asked it to do, and it ran to completion. And so this step, because we're interacting with this, kind of outside space, at the very beginning, when I was saying I want to make access available to this project, this is why, maybe an hour and a half later, why it was important to choose that setting all the way at the beginning, because it allows us to be able to find this file from the GTEx website and pull it into this Cavatica space. So now I can do ls, and here's my lymphocytes file. So I can unzip it and do ls again, and then we can do head. And you can see that it's kind of this long list of expression values of our different GTEx data with the gene ID, and then the gene name, and then the the individual GTEx samples. So you would want to pair this with additional GTEx data as well, so about who are these samples. Again, so you'd want the GTEx metadata, which describes who are the participants, where did these samples come from, in order to be able to choose which ones are most interesting for your analysis. But again, that goes a little bit beyond kind of the intro level demo that we're showing today, but just kind of show you where you could get this additional data from, how you can pull it into a particular site. So there's two more things I want to show. So one other thing I want to show is how else can you get data? So data for GTEx is also available in another NIH platform called the Anvil, which I can show on my screen. And so I'll copy the link here. I'll pull this over here. Actually, I believe I'm done with my RStudio. So for the sake of showing good cloud computing technique, I'm going to say I'm done with this. So I'm going to turn off the lights when I walk out of the room. So I'm going to stop the computer and confirm. And so that will save everything that I just did. And so if I want to go back, oh, shoot, I needed that graph, I can log back in and turn it back on, and I can get that graph again. So that'll be easy. So that session was 10 minutes, and that one cost me 8 cents to run that. Okay. So the GTEx the data is also on the Anvil, which is another NIH program site. And so we can log in with our NIH login, really similar to the Kids First portal. And we can look for, so they have a couple different data sets in here, but particularly interested in this GTEx data, Common Fund GTEx. So that's what we're interested in. So this has all of our information for all of our different GTEx participants. And we can look at sequencing data, and if we're interested in RNA-seq data. And we want files for these participants. So we want GTEx, and we want the, for example, if we can choose allele-specific expression. So it gives us this kind of allele-specific accounts, but we can also choose our transcriptome profiling. And so it would give us our unaligned reads if we wanted to go all the way back, as well as our expression files. And so then from here, you can then generate a manifest, and you can export all of these to 7Bridges platform, which is our Cavatica, export to Cavatica using this button. And so it's going to take a while because I didn't narrow it down very much further. So there's 54,000 files, but this will download a manifest that we can then upload into Cavatica. And it will then pull all of these files into our project. So I'm not going to show that today because that's going to take quite a bit of time. But that's another way that you can access kind of the raw files for GTEx and bring them into Cavatica in an interoperable environment without having to do that copy, that wget that I showed before. All right, so there's 8 minutes left. And so with what I want to do today, I want to kind of bring us down for a landing and then set us up for our course tomorrow. So I'll show one more thing. And so if I go back to Cavatica, and I go back to my task. So today I ran this demo of this DE-seq2 run, and everything ran, to completion, which was great. And so to share, peek behind the scenes, sometimes on these demos, the instructor makes it look very easy. You click these buttons and everything works perfectly the first time. And the reality is that doesn't always, work that way. And I just want to be transparent with folks that, especially if you're getting started in bioinformatics, working through error messages is a really big part of getting started and can be a really frustrating part. And so if I go back to my main tab, which shows all of my recent analyses, you can see I was in here, the past couple of days this week trying my DE-seq2 run and getting a lot of failure messages. And so eventually I got it working, and I'm really happy that it worked today to be able to demonstrate for you all. But failure is part of the experience, I think, especially when you're using a new tool and especially when you're getting started for the first time. So how do you go from failure to completed? Two things, my tips that I'll share, I guess three things, is that one is that if you're having trouble, definitely. So if you have a task that fails, it can give you some error message, but the error message isn't always intuitive. So sometimes you have to dive into those logs, which provide a more useful message. So if I view the logs and I view the error, it gives us some more specific information that we can use to dive in. So troubleshooting tip number one, check the logs, find the error message, try to figure that out. If you don't know what the error message means, ask for help. So Valsera offers office hours twice a week in order to help you kind of troubleshoot some of these problems. If you click that, that blue question mark and paste that error message, help, I'm having trouble, this is what I got, folks can help you. You can also Google search that error message, see if other people are getting it, try to interpret, translate what does this mean in English in terms of like, how do I solve this? What do I need to change or fix to be able to get this to work? So there's ways to ask for help. And then the last place that I'll emphasize is really make sure that you're reading and understanding, what these software tools are doing. And so on Cavatica, a lot of the software tools are really well written. So this one's written by their, software team. They didn't develop the DESeq2 app, but they did write this, build it in the Cavatica environment. And so they give, this one is really good because it gives you really clear explanation of what you're looking for. And this helped me troubleshoot and realize this is how I knew I needed to patch that metadata and add that sample ID by, just reading the description and a little more carefully and thoroughly. So always try to be conscious of, if you're using the tool and there exists really strong, powerful, readmes, be sure to check that out as you're going through. All right. So with that, I will kind of bring us down for a landing today. So we're right at the 2-hour mark. So I'm going to go back to my PowerPoint. And here we are. So we completed our analysis walkthrough. And so let's see, let's pull up on the full screen again. And so hopefully this was a good chance that we showed you how to, the kind of the concepts involved in RNA-seq. I showed how you can select files on the Kids First portal, and then how you can run this DESeq2 analysis on Cavatica, and then open that R data file in the Interactive Data Studio. And then I showed a couple different ways that you can incorporate GTEx data into your analysis by pulling from the site using a wget or by navigating to the Anvil and doing a download that way. So then tomorrow, like I said, we've been scaling and building in complexity. And so today might have felt like a big jump, from the lesson to now we're doing the analysis. And so tomorrow we're going to jump a little bit higher. So Sangeeta is going to lead us in a slightly more complicated method. So instead of using this existing DESeq2 app, we're going to learn, well, how do you build your own app? If I have my own workflow, how do I bring that into Cavatica? And so Sangeeta is going to demonstrate, if you have your own tool, your own analysis that's not shown in that app store, how do we make it, possible for us? And so that's where we're going to tomorrow. And so with that, I think I'm going to close for today. If you have any additional questions, let me know in the chat. Happy to try to answer any final thoughts. But otherwise, we can wrap it up for today.
Hi, David. I had written a question on.
Hey, Harlan. Yeah, I see. I'm reading through it right now. I'm trying to use this new database to find control files. Another question says proband imply definitely diagnosis of the disease of this individual. Yeah, great question. So as far as the control files, so Kids First doesn't support control files on our own. But we do this is where we make GTEx available because these individuals are not necessarily diagnosed with conditions. And so I would suggest GTEx as a possible source. As far as does proband imply definitely diagnosis of the disease? So yes, so proband for Kids First means that it is the participant that starts, it's usually it's the child and they have the condition in the particular study. And then they have parents as well. And so it's possible that parents might also have the condition, for example, related to like our cleft lip studies. So sometimes a child will be the proband that is born with a cleft lip, but maybe that child's father also has a cleft lip. And so that father would not be the proband because he's not the child, but he would be labeled as, I think it's affected status equals true. And I can share my screen and we can show. Let's see here, do this, and then we'll pull this back up. So we're talking about kind of phenotypes and conditions. So I can show participant, or excuse me, it's under clinical. And so we would want observed phenotype. And so this would be where you would choose a particular phenotype. But then probands, not all people with phenotypes are probands. So if I choose, I'll choose oral cleft because we have a lot of oral cleft data. Apply. And so you can see that many of the individuals with the oral cleft are probands. Proband is true. But for some individuals, so this proband is false. So this would be probably somebody's mother because they're a female individual and they're not a proband. So they're probably the mother of another participant who is a proband. But they were also born with a cleft lip. And so that's what that kind of proband status is. But if proband is true, you would expect them to have observed phenotype, or you expect them to have the condition of the particular Kids First study.
Thank you.
All right. Just to clarify, all the information about how the VCF files in the data portal were obtained are in the Kids First DRC workflows in Cavatica. Yes, that is true, Monica. And so you can access those if I pull up Cavatica and under public apps, workflows and tools, and you type, we can browse the apps. And then if you search for KFDRC, KFDRC, there we go. And this provides all of our Kids First alignment. So for example, for the alignments, you would want to read about the alignment workflow. For the VCFs, you would want to learn about the joint genotyping workflow, and then you can read through the documentation there. All right. Well, with that, I think we will wrap up for today. But thanks so much for joining. Thanks for sticking with us on day 3. And again, we're looking forward to tomorrow when we'll, again, slightly more complex. We're going to build our own workflow. So it should be an exciting conclusion of our course here. All right. Well, thanks, everybody. I hope you all have a good day, and we'll see you back here tomorrow.